just make sure you come talk with me and we can go over the exam. All right. So I think we left off here, kind of hard to remember where we were, but I think we were talking about biofilms. And so I don't know why my computer is freaking out already. Let's try that. All right, so realizing that microbes, when they live their normal life, they tend to form biofilms, at least for a good stretch of that. They can come in and out of it. It's not permanent. So they can form biofilms when it is strategically a good idea. And then when it's not, they can break loose and disperse and find another habitat. And so it's more dynamic than we initially thought. And we can find biofilms all over the place, including inside your body. And so I think we left off talking about the idea of the difference between, biofilms in a hot tub versus a sauna environment. And the idea that even if you clean the hot tub where you sit, that area, the actual tub, you can clean that really well and disinfect it. But if they don't think about what they're doing for the pipes, very often biofilms form in pipes, not just for hot tubs, any pipe. City pipes here, the people that do the wastewater treatment plant, and they, just getting biofilms to stay at a minimum inside pipes can be challenging. Hulls of boats, you can find biofilms there too. So lots of biofilm environments for sure. And the hot tub in particular has been more problematic in the sense that biofilms can persist even with cleaning and can cause human disease. In this case, I talked to you about that person going blind in one eye after sitting in a hot tub because of the aerosols. I think most of you have experienced a hot tub perhaps, and you see those aerosol bubbles coming up. And that's what they think happened, is that might have been part of the issue, that the bacteria were on the aerosols, the droplets, the fine water droplets. So there was another case study where it was two elderly women were on vacation and they went to a resort and were they didn't even go in the hot tub. They just sat on the edge and dangled their feet into the hot tub. And they both contracted genital herpes. They went to the doctor and the doctor's like, did you have unsafe sex? And they're like, we didn't even have sex. So, like, how did this happen? And then they did the, epidemiology of, okay, where have you been? And they tracked it down to that hot tub at that resort. What happened was, before those two ladies went to sit there, a young gentleman was there with active herpes outbreak and sat there. And the virus persisted long enough in just right where they sat. So it was incredibly unlucky pick of where they chose to sit. But, these case studies are, being an epidemiologist, like the ultimate mystery solver, because it can get, we'll talk more about it later, but my hat is off to epidemiologists because that can be very, very challenging. So anyways, hot tubs, be mindful, be careful, stay healthy. And saunas would be my option rather than a hot tub. I wanted to try to do something relaxing because you usually, when you go, especially into a public sauna, you should anyways put down a towel to sit on. And usually a lot of public saunas, you have a bathing suit in addition. So a little bit cleaner, drier heat, biofilm production is not quite so high. So next week, in lab, you'll do some experiments looking at microbial growth at different temperatures. And we're actually going to provide one that mimics a sauna temperature. So we'll see how some of those microbes fare under those conditions. So sauna wins, thumbs up. Has anyone here taken saunas? Couple. Does the PEEF saunas, do those still work? For a while they had them shut down, but I'm glad they're up and running again. So they can be very, very helpful. My grandfather was 100% Finnish, and anytime he started feeling off, he had a sauna in the backyard, and he would just fire that up and get it really warm and go sit in there. And he swore it helped him deal with whatever was trying to make him sick. So biofilms are dynamic. you have lots of community social interaction going on between the microbes, and they actually refer to some research. They actually use the word socio-microbiology because they now know that microbes are communicating with each other in a variety of ways, one of which is quorum sensing, which we'll talk about soon in more detail. But they find many microbes do some form of communication, and the best studied examples are quorum sensing form of communication in order to coordinate their efforts to build a biofilm. And then they live in that biofilm together, and often the biofilm will actually, of course you have to remember microbes are tiny, but they've done experiments in the lab where it is a three-dimensional thing. Like it kind of reminds me of downtown Chicago, where you see aspects of it where there's kind of skyscraper biofilm production, and then you have like streets where the water flow goes through easier, and tunnels, and very, very interesting three-dimensional design to these biofilms, even though they're very tiny. But some of the imagery is really quite fun to look at. So they tend to form in areas where the microbes come along. If they're mobile, let's say, they find a patch where nutrients are super abundant, and they're like, "Ooh, let's set up camp and take advantage of this." And so they'll stay there for a while and use up the nutrition in that area. And then when things get too depleted, then they'll say, this isn't such a great spot anymore. Let's pull up the tent stakes and go somewhere else. And so they can go back to being, more free living and exploring their environment for another nutrient patch. And so, or, if, well, we'll talk about the human body in more detail later. I'll save that for later. But it's often, if you study natural biofilms, they're mixed species, so it doesn't all have to be the same species, so they can get along with each other, and they can coordinate their behavior to capitalize on that. In this case, I'm describing a case where, it's a really nice nutrient area where they're getting access to really good nutrients. So here's a nice visual, including real imagery on the top where they're showing, this is Pseudomonas. Yeah, in the top image where they're trying to show you the three-dimensional architecture of biofilms, and then the drawing below it trying to show you the step-by-step stages. And so planktonic just means free living, swimming around, like you imagine a stream or a pond, and then maybe they happen upon a log in the water that has a bunch of nutrients, so then they settle on it and start communicating and then start doing the biofilm production. And then they're trying to show that three-dimensional architecture as well in the drawing there. So they have devised these little pieces of equipment in the lab where you can do these biofilm studies and look at the progression, and they get these types of images spit out when they are running experiments. So really does kind of look like a city, where you have, skyscrapers and then streets and tunnels and all of that. So it's, when they started looking at this more carefully, I think it was kind of a surprise that most microbes do biofilms. They thought it was kind of a special thing, but they realized that it seems to be a strategy that most microbes can get into. And again, this is showing step by step, and then some image down there of an actual biofilm with the different species stained different colors. So trying to get at the idea that it doesn't have to be the same species. Here's another one. So this is Staphylococcus, let's see, bacillus, a bacillus species, Bacillus subtilis, is green, and Staphylococcus aureus is red. And in this particular study, they were demonstrating how the presence of B. subtilis was protecting Staphylococcus from a disinfectant treatment. And so this was on a piece of equipment used in a hospital. And so somehow Bacillus was helping staff survive in this by this biofilm production. And then on the right, this black and white image here on the right is they applied, I believe, the biofilm edge. The biofilm is from a washing, disinfecting piece of equipment. And so they're showing how the disinfectant, when bacillus was present and they were in this biofilm, staph could survive the disinfectant. And they're not worried so much about B. subtilis. That's not really the problem. But Staph aureus, that can be a real problem, especially in hospital settings. It can often be the methicillin resistant type Staph aureus that can cause a lot of problems in patients. So biofilms can be very handy that way. It can slow down diffusion of chemicals in this case, where it's not really impacting the heart of the biofilm. It can also, if you take antibiotics, slow down the diffusion of the antibiotic to the actual cells. That was one of the questions on the exam. And a lot of you mentioned biofilm production, which is a good strategy. So this is, again, just trying to show you some different ideas of why they might want to do it. Certainly to help protect them. They can get good nutrients. And they can also, it's a good spot for horizontal gene transfer. So it is a kind of a hot spot for swapping genes. It can really help increase their ability to adapt to certain environments, depending on what genes they got access to. So biofilms, they can be a problem from a human perspective because they can create situations where there's really luxuriant growth of microbes where we don't want them. If you think about hospital equipment, for example, catheters that they insert in patients often can get biofilms that are very challenging to deal with and very quickly. And so that can be a problem for the patient, leading to all kinds of infections that they didn't start out with. Like they came into the hospital for a different reason, and then they end up with an infection due to a biofilm catheter that forms as it's inserted in them for any length of time. So, they're starting to look at how do we try to knock down biofilms or eliminate them in certain cases, like in a hospital environment. So they're actually looking at bacteriophages, viruses that attack bacteria. They're calling them smart bombs, and using them to break up or break down biofilm. And so top and lower image, the lower image was a biofilm that got treated with phage. And you can see a huge difference in the amount of microbes. This is Enterococcus faecalis, which used to be called Streptococcus faecalis, which is a nasty human pathogen. And so this idea of knocking down the biofilm production can be very useful, something that we really do need, especially in hospital settings. It can be amazing how quickly biofilms can form. Another study that was done, anti-biofilm peptide. And so this is just the treatment of Pseudomonas biofilm. On the left, it's untreated without the peptide being mixed into it. And on the right, you can see a huge difference in the amount of Pseudomonas cells once they did treat it. So they're looking at different strategies to try to safely knock down some of these biofilms. And another option that they're exploring, but they haven't really quite got there yet, is quorum quenching. So this was published a few years ago. And we're going to get into quorum sensing and quorum quenching next, actually. But this is the idea of stopping the bacteria from communicating with each other. And if you can stop the communication, the idea is you'll stop biofilm formation. Because first they communicate, then they do the biofilm. So if you can stop the communication, you can stop the biofilm. That's the leading hypothesis. So they are working on that, but I don't think they've reached any successful, like it's not in implementation phase yet that I'm aware of. So this is just for you to watch on your own. It's an active link. If you just click on the PowerPoint when you're studying, it'll show you a short video on biofilm formation from start to finish. So if you like the idea of a video, that's an option. So we'll talk a little bit more about biofilms here and there through the semester, but I also wanted to finish up this section on growth by looking at some other unusual ways microbes can grow. And again, it's very dependent on their habitat. Like what are they faced with? What kinds of challenges and how they respond to that. And so they can behave in very interesting ways. So we've already talked about endospores, how amazing those structures are with the gram-positive bacteria. And I mentioned a little bit about heterocysts of cyanobacteria. Again, heterocysts are a very good structure for these cyanobacteria to mate, so that they can go, and do their necessary things with the heterocyst is to fix nitrogen in a special structure. And so again, it's just a way for them to adapt to their environment to create a mechanism where they can fix nitrogen in the presence of oxygen. So I'm not going to get into it in huge detail, but basically any bacteria that fixes nitrogen, doing it in the presence of oxygen is very challenging. And yet cyanobacteria are aerobic bacteria. So it's a real conundrum. It's like, I need to fix nitrogen, but there's all this oxygen. So cyanobacteria solve that problem by creating a very oxygen-free heterocyst, where they fix nitrogen there and then share it with the other cells nearby. Fruiting bodies are another one I'll mention. I'll show you some pictures of really cool fruiting bodies and how they go through a very specialized differentiation process to do this. And then I'll mention a little bit about Streptomyces bacteria and how they differentiate, which is in some unusual ways for a bacteria. So I'm not going to go into this in detail. I'm just reminding you, we've covered endospores. So that is a cell differentiation idea. There's a very special structure that gets formed through a very rigorous process on behalf of the bacteria. Here is the heterocyst idea, cyanobacteria, where you can see a string of cells here, but only one heterocyst. So they fix nitrogen there, and then the nitrogen gets diffused out to the neighboring cells so they can all share in the bounty of some lovely nitrogen in a form that they can use. And so you have to do it in the absence of oxygen. Fruiting bodies are fun. So I'm not talking about mushrooms here. I'm talking about bacteria that are forming their own fruiting body by coming together in huge population. So what they're showing here, I think they used an agar plate study for this, where they inoculated with some Myxococcus xanthus cells. And then what you do is you create, normally when they are studying this in the lab, they try to create starvation conditions so that they start to swarm together. And they're all like, hey, we're running out of food, let's go find some food. And eventually they'll go, uh-oh, we don't have a bunch of food anymore, we're starting to starve. And that's the trigger to start getting formed this fruiting body. That is a mass of these cells. So they're all cooperating, coming together and cooperating to form this fruiting body. And what it does is it allows some of these cells to survive, because some of the cells will form more resistant structures, their own special kind of spore, and it allows them to go dormant. But not everyone will do it. That's what's kind of weird, is that some of them, especially the ones lower down, that are attached to the substrate, they won't go into a dormant phase. They'll eventually just die off. And so some of the cells make it, they'll go dormant and survive, but a lot of cells don't. It's kind of an interesting, again, sociomicrobiology. They actually start using terms like altruism and that kind of thing. So it's kind of interesting to think about how these consortiums of cells, some survive and some don't, but they all come together to make sure at least some of them will survive. So here I draw a line again. So they call it fruity body. So get this collection of cells forming very special structure, and then just some of them will form these dormant spores that will allow them to wait for better times. And once a better situation comes along in their environment, they'll get active and grow again. So yeah. Right? Very good question. Right. You would think, why don't all of them form spores, right? And so the thought is that some of the cells have to attach to the substrate that's their job. Some cells have to build a column up on the substrate so that the spores that form have a better way of dispersing. And so it's this weird kind of differentiation of labor or jobs. But ultimately, what that means is the cells and the ones attached to the substrate will die. And only the ones that are raised up high make it. And it's like, who decides that? You know, like there's all kinds of communication going on. Form sensing again is involved in this, where they're talking to each other, and it's like, well, someone's got to be the root, for lack of a better term. Someone's got to be the stock. So it's very interesting. That's why they started using terms like altruism, because altruistically, you know, these cells down here, they're helping with the final goal, but they don't get anything from it directly. However, their species gets into the next generation. It survives. It keeps going, so to speak. And so the fact that these cells hopefully will disperse into a new habitat that will allow them to survive and thrive and keep growing. And so it's a way not to go extinct, essentially. But that's what they're coming up with in terms of why that odd differentiation of labor goes on. So really fun to think about. And when you look at them and they stain them the right way, they're quite beautiful, but they're super petite because these are bacteria. So it's not like you would stumble on this with the naked eye out in the woods or something like you can with fungi. But studying in the lab, they can be really fun to look at. These have been specially color enhanced. So myxobacteria have been studied for a long, long time because it is such a, from a human perspective, it just seems kind of an oddity that some cells are sacrificing themselves. They're just part of the structure to help the cells that will go dormant. And then Streptomyces bacteria are bacteria that I studied for my grad work, postdoc work. And I really enjoy working with them because they're very atypical bacteria. They're not like E. coli or Bacillus that you studied in class because they look like fungi. They look like petite fungi under the microscope in that they form hyphae like fungi do. And so they'll grow out in a very hair-like and thread-like appearance. And if you have a Streptomyces bacteria under the microscope next to a fungal hyphae, you can immediately see the size difference. That's why I say the Streptomyces are very petite, because their hyphae are very slender compared to fungal hyphae. But it's still that we still use the word hyphae. We talk about mycelium, just like we're talking about a fungus. And so also when you look at their colonies on an agar plate, they don't look normal, like like what you've seen, like E. coli and Bacillus. They actually tend to, well, they change over time. So first, the colony will look kind of tan. It won't look wet at all. It looks very tan and dry. And then it starts to go white and grainy, almost like powder. And then that white shifts to some color. And here you can see in figure B, that blue, that's like the real color. That wasn't photoshopped. Those are spores that are being produced. And all those dark beads on there, that's pretty much pure antibiotic that has exuded out of the hyphae and then crystallized upon exposure to air. And I see that all the time with the streptomyces, especially from the soil. You can see them exuding the antibiotic like that. So their colonies change over time and can form some very beautiful colors. And then under the electron microscope, all that blue area, those are the spores. So they tend to form pigments right as they're sporulating. They tend to form antibiotics also when they're sporulating. As they're shifting into the spore state, they pump out a bunch of antibiotics. And so, again, very kind of step-by-step way of how they grow and then differentiate into these different spores. And I love working with them too. You probably noticed when you're in lab that some of the bacteria you work with, they don't smell the best. And streptomyces bacteria, they smell like wet soil, which I like that. Like after a rain and you go walking in the woods and the soil is nice and damp, that's what it smells like. And so for me, that's much better than smelling a plate of E. coli or something like that. So here's the step-by-step of their growth form. So just trying to show that you get this progression of, you know, the spore will germinate, grow out the hyphae, and then you get usually starvation will trigger them into forming what we call aerial hyphae, substrate, and allows them to get up on it so that they form their spores that are hopefully high enough in the air current, so to speak, that once those spores break free, they'll get carried off by a puff of air and land somewhere suitable. So kind of analogous to what we see with fungi in many ways, but these are definitely bacteria. And most of you have had experience with streptomyces already in your life if you've taken antibiotics. Almost all the antibiotics that we use are from the streptomyces. And so that's another reason why I wanted to study them, because I thought they would be good biocontrol agents to help plants defend themselves against pathogens. So this is just kind of a summary slide showing the options for the ways that bacteria in particular can go through some different growth forms, almost to the point of, you know, when we say multicellularity, we're talking about lots of these cells coming together and forming a structure of some sort. And so very different, interesting way of living their lives. Okay. Could go on and on, but I think we'll leave it there for bacterial growth. So the next topic on the syllabus is looking at we're going to get into so I have it down on the syllabus as regulation of gene expression, but I'm not really doing that per se. Probably rename that. So the next topic I want to get into is looking at gene expression in a way, but in a very focused manner. So let me pull that. What I want to get into, because I realize when you take BI-218, you do a fair bit of gene expression information in that class. So I've cut back on that, and I'm zooming into a couple of things that are unique for microbes. And you probably don't get a lot of maybe some of the microbe stuff. So I'm going to focus on quorum sensing and nanotubes and a couple of other ideas of how microbes are shifting their gene expression. So I'm looking more at what might cause them to alter their gene expression and then how do they do that. And so this is here in America, pretty much currently the two pioneers are Dr. Bonnie Bassler and Peter Greenberg. We've done a ton of research on quorum sensing. Dr. Bassler tends to do Vibrio cholerae, cholera, and Peter Greenberg works in several different types of bacteria. I'm going to post I haven't already, I'll double check, in the video section of this EDUCAT page. There's a TED Talk with Dr. Bassler, that I'd like you to watch. Very engaging speaker. I've seen her talk at microbiology conferences, and she's very fun to listen to. So I think it's like a 20-minute video that I'd like you to watch. So, and it's all about the basics of quorum sensing. What we're finding out now, the more we look into quorum sensing, how much more complicated it is than we thought, which is pretty much seems to be the way with biology and science in general. Once you start digging into it, you realize it's a little bit more complicated than what we initially thought, but that's what's so fun about research. You can never get bored. And so I want to start with quorum sensing. So this is the idea that microbes don't live in isolation. Even if if it's just two E. coli cells next to each other in a colony, which remember, one colony of microbes on an agar plate that you've looked at is actually millions of cells all hanging out together. And so you can have, two E. coli communicating with each other, but you can also have, we now realize, diverse species able to communicate with each other. It's not just related species. And so this idea that, of course, they don't talk, but they use chemicals. And so this idea of they have to have some mechanism in place where they're passing chemicals around and others are able to pick up on it and understand it and then behave differently in response. And so this, you'll see papers that are published where they talk about autoinducers, sometimes they use the word pheromone, but all of that is dealing with this idea of quorum sensing, autoinduction. You'll see that phrase a lot. So autoinduction and autoinducers, the autoinducers are the actual chemical. They say autoinducers, self-inducing, because they're trying to point out that the bacteria are making these chemicals and then they excrete them out. And so they're autoinducers. And plus, they also ramp up it. They kind of do a feedback loop on their own physiology to ramp up certain things out in their own physiology. So it's kind of a feedback loop in some regard. So this image is trying to show chemicals getting released on the far left, and this idea that you need to have some kind of receptor. So this purple and green stuff here, those are protein receptors that the chemicals will dock into, and then that sends the signal to the internal parts of the cell and ultimately to the DNA. And then you'll get a shift in gene expression. So here you see some ideas: virulence, bioluminescence, other group behaviors. So that phrase I really want you to appreciate. This other group behaviors, because I mean, that's kind of like the whole point. They are doing this because now they're in a group, they can take advantage of that. And so I'll work through this process and you'll see what I mean as we work through it. But this group behavior idea is very, very important that you key in on that. So here's a cute little squid in that image. And you may have learned from some of your other classes, they're just watching documentaries, that some squid can bioluminesce. And those are bacteria that are causing that. And some fish can do that too. You tend to see, fish or squid that live in deeper depths where it's dark a lot of the time. They'll be able to bioluminesce in some way. Figure C and D there, that is the Vibrio. And so if you just look at a plate under the fluorescent light, it would look like what you see in figure C. It doesn't look anything special. But if I told you, hey, take that plate and go into the walk-in incubator in our lab and just shut the lights off in there so it's total dark, you would see what figure D is showing. And you don't need a black light or anything fussy. Just let your eyes adjust and it will start to glow like that. And so they're doing bioluminescing because each colony, remember, is millions of cells. So they are quorum sensing with each other like crazy in those colonies. And that leads to quorum sensing behavior. So they first figured it out in Vibrio that hangs out with squid. And so that's a very special situation. So they thought that was unique, like only Vibrio can do this. And well, I mean, it is pretty special to bioluminesce, but I'm talking about quorum sensing. Like maybe that's just special for Vibrio and its ability to bioluminesce. And then they started looking at other bacteria and they went, oh, wait, it seems like other bacteria that don't bioluminesce are also doing quorum sensing. What's that all about? And initially, with the initial research, they said, oh, well, gram-positive, they do X, Y, and Z when they quorum sense. And then other researchers said, oh, but wait, gram-negatives do A, B, and C when they quorum sense. So they started realizing that there's some different strategies. Now, the further that this research has progressed, we know it's not quite so clean cut. So we're finding that there's some overlap, but I'm going to maintain teaching it this way because it's the easiest way to think about it, I think, for learning it from scratch. So like I said, Vibrio fischeri, which by the way has been renamed L. Vibrio fischeri, but this one's gram-negative. And like I said, it's symbiotic with the squid. And so they started working out how gram-negatives do it first. And they did a lot of lab-based studying, of course. And so they isolated Vibrio from the squid and started working with it and trying to track down this whole process of how do you get them to bioluminesce like that. And you can get them to do it separate from the squid. These are Petri plates. And so they're like, oh, okay, the squid doesn't have to be part of the equation here. We can separate them out, still make them do it. And so they started working with them in the lab, which was nice. You didn't have to worry about growing up the squid. You could just work with the bacteria. So when they looked at Vibrio, they realized it was making this chemical. It was an N-acylserine lactone, which you can abbreviate to HSL or AHL. Being shown there on the lower left. So there's the lactone ring. That this part is the lactone ring. Okay. And then the acyl side chain coming off of it. And it has a nitrogen group there too. So that's the basic structure of an acyl homoserine lactone. And then you can see this acyl chain. Well, actually the left picture shows it. They show an R group at the end of that acyl chain. That just means there's variation on that chain. Like how long does it stretch out? How many carbons are attached? Okay, so that can vary, but the lactone and the homoserine and the oxygen, that all stays consistent. All right, but then the side chain can vary a little bit. So it's this chemical signal that they're making. So they've synthesized that in their cells. So it's self-produced, auto-produced. Then it gets excreted out into the environment. So you have to imagine a bunch of Vibrio in that colony on that agar plate. Remember, each colony is millions of cells. So as those cells are building in their population, as you're incubating that plate, they're all producing at low levels homoserine lactone. They're all excreting it. And once you get enough cells all doing that in an environment together, then it will accumulate to a high concentration of that chemical. And it will then follow the concentration gradient and diffuse back into those cells. So they often say there's a threshold level of the chemical. There's a threshold level. And then once that gets reached, you get diffusion back into the cell. And then what it does is those chemicals find a regulatory protein inside the cell. And then that chemical binds to it. So now you have the regulatory protein, the chemical bound to it, and then that goes to the DNA and will turn on or off certain genes. So we just kind of say it alters gene expression. It could be turning on, it could be turning off. So alters is a better way. Induce or repress, if you're more of a molecular biologist, you would say it induces gene expression, turns it on, represses, turns it off. So that's the basic idea in gram-neg. So it's fairly straightforward if you think about it. You have this idea of the homoserine lactone getting made and getting excreted. So that's what they're showing here. And then it gets excreted out. of the cell, and then you have to imagine a bunch of these cells all hanging out together, all doing this. So then you'll get a threshold level, say that word right there, threshold concentration. And then by the laws of diffusion, it diffuses back into all the cells. And you'll get the binding, in this case, the regulatory protein is LuxR, but there's different regulatory proteins. It just depends what bacteria you're talking about. In this case, it's LuxR, and then it binds to that, and then they're trying to show that it will kick on, in this case, gene expression for bioluminescence. So in this particular case, it turns on gene expression. So when Vibrio is hanging out, whether it be with the squid or on an outer plate, it's always producing this quorum sensing signal at low levels. So it's not a huge cost. It's just like, I'm going to make a little bit of this and send it out. And it's kind of like their way of feeling out the situation. Like, what do I got around me? How many other Vibrio are around me? And so the reason they call it quorum sensing, if you've ever belonged to a club or a committee, you have to have so many people present, a quorum, in order to pass a vote. So that's why they came up with quorum sensing, because it's a way for, in this case, it's a way for Vibrio to say, well, I really only want to do bioluminescence if there's a lot of us around, right? Would you want to do it if you're all by yourself? I mean, you wouldn't give off very much light, wouldn't be very effective. So that doesn't make much sense. I don't want to give off light all by myself. That's not going to get me anything in return. But if there's a bunch of us doing it, and especially if they're with the squid, it might provide a mechanism where it's a payoff for them. And so this is a way for them to wait, figure things out, how many are around. I don't want to really do this until there's enough of us to really make an impact on our goal. What do you think of Vibrio hanging out with a squid? It's a kind of a win-win. It's a symbiotic relationship. The squid gets something, but so do the Vibrio. Can you think what Vibrio might get hanging out with a squid? Another thing you might want to think about, the squid actually recruits these Vibrio, like it can pick them up, so to speak, in the environment. The fish do that too. Like they'll recruit Vibrio. And so now Vibrio are hanging out in if you've seen those lantern, I think they're called lanternfish, where they're in the eye socket area of the fish, and the fish blink and can flash light. Have you seen video of that? And so if you watch the video with Dr. Bassler, when you watch that, she'll explain a little bit more about why this would be a good thing for the host to partner up with the Vibrio. And it can be a good thing for the host, for the fish, the squid, but also for Vibrio. If you are recruited by one of those organisms, that's a pretty sweet life. You get to hang out right in the tissue of a host that can feed you, rather than in the open ocean where you have to hunt for food all the time and pretty dilute food. Now you're in a very concentrated area. You're in the tissue where you got nutrients leaking from the host all the time. If I were Vibrio, I'd definitely want to hang out with a squid, because you get a protected spot, food all the time, and all you have to do in return is this, and they'll keep you. Pretty sweet. So something to think about when we'll get more into why are they doing this, but let's first learn a little bit more of how. So here again, this is trying to show that you have to have a threshold level of cells in the area, which means there's going to be a threshold level of the chemical as well. And so they're just mapping out the growth of Vibrio in this case. And then they're showing that it's not until you get high population numbers, high number of cells, and a high number of chemicals, that you really see the bioluminescence take off. And so you have to have great enough numbers of the cells and a high enough concentration of the quorum sensing chemical. And then if you get all those conditions met, you'll end up with turning on the genes necessary for bioluminescence. So this, again, when you're studying, I think this is still an active link. You can click on it when you're studying, and it gives information on the process of quorum sensing if you prefer the video. So this idea of these homoserine lactones, they thought initially, oh, there's just like one chemical, and now they know, but wait, there's more. And so here we're looking at all these different organisms, and you're looking at their version of homoserine lactones. And so you can see that they're trying to show that anytime it says R, so they're saying the R group could be a methyl group, or it could be, expanded quite a deal. And so you can get a really long side chain or really short basal side chain, and different microbes can do it. It's not just Vibrio. So these are all gram-negative. But you can see that the basic structure of the homoserine lactone is the same. So it does make you wonder, well, if they're chemically related, can they understand each other? Like, can Pseudomonas, when it produces its homoserine lactone, can maybe Proteobacterium here, or maybe Chromobacterium, can they understand it? And so it's kind of like foreign language, like French versus German, can there be some understanding here, or is it totally no, they can't figure each other out? So we'll talk a little bit more about that. But the more they look for gram negatives, they see lots of variation, but it's really only the side chain that varies. The rest of the structure stays the same. So it really does make you think about, is there any kind of crosstalk that can happen? So we'll look into that too. And then this is just trying to get at the idea that you have to have a group of cells together. One cell living all by itself, it doesn't make sense to go through all of this. But if you have a group of cells, then you can get a threshold population, a threshold concentration, diffusion back into the cell, which then finds that should just be regulatory protein, which can then alter the DNA gene expression in some way. So that's what they're finding for gram negatives, a very basic idea of quorum sensing. So on Wednesday, we'll get into gram positives and then a little bit of the reasons of why would they want that. Okay.